Continuing on with our discussion about membranes, we need to figure out how does transportation work across the membrane? Because your cells are constantly intaking and outputting different types of ions and particles and molecules. How does this all work? How do they get in and out through the membrane, through that phospholipid bilayer? Well, the first idea is the actual membrane is selectively permeable. It's only going to allow certain things to travel through without special adaptations. Now, special adaptations could be channels or various other components that help, but some things simply go right through the membrane without any special help. Now, these processes of going across the selectively permeable membrane can either be passive transport or active transport. In the case of passive transport is going to not require ATP directly. That means it's not directly requiring energy. While the active transport will require an energy source. So if we look at passive transport, we'll start with diffusion. Now there are different types of diffusion, but simple diffusion is just the most basic one there is. Simple diffusion is simply utilizing kinetic energy, the energy of movement. You don't need to put any ATP in. There's no energy needed for this. These that are going across the plasma membrane through simple diffusion are just passing right on through. You can even think of simple diffusion in a non-membrane situation. If you ever had a little color tablet and dropped it in some water, you can watch that color start to spread from that one point and spreads out. That's diffusion. From a point of high concentration, moving out to points of lower concentration. So simple diffusion can go directly across the lipid membrane from a point of high concentration to a point of low concentration. So you can see in this example, we'll start with the time scale goes from the white all the way to the red, so starting to end. You'll notice that initially, this area doesn't have any of these molecules. That's because it's a low concentration. But as we move down the time scale, as the time elapses, you'll notice that some of these have come right through the membrane. That's because they're diffusing, simple diffusion, high to low concentration, right through the plasma membrane. By the time we're done, both sides, the side that had the higher concentration initially, and then the side that initially had the lower concentration, now both sides are roughly the same concentration. Simple diffusion ends when both sides are at roughly the same concentration, because that is homeostasis. The two sides are balanced. Each side has about the same concentration, so there's really no need for one side to give more to the other side. There still might be some back and forth, one ion moving here, one ion moving there, just to maintain that. But the bulk of the movement is finished once the sides are roughly equal. Next type of diffusion is facilitated diffusion. Now, in the case of facilitated diffusion, you'll notice it's talking about transport of glucose, amino acids, and various ions. The idea facilitated here means something needs to help out. They can't simply go across on their own. So we're looking at protein carriers or pass-through channels, something that is going to be in the membrane to help these compounds, these particles, move through the membrane. So the top example here you can see is simply a channel. The ions go into the channel and pass on through. That's a way for facilitated diffusion. Without that channel, those particles could not go through the plasma membrane. So it's a helping aid. The bottom example you can see is again a channel, but this one, if you notice, is going to be closed at the bottom to start with. That closed component opens up once the actual molecule is inside. So it takes things through, brings it in, and then opens up the other side. Takes it in, opens up the other side. The idea is it's not a free flow, but it's kind of a mechanism that allows them to pass through as it goes this way. So again, protein in the membrane, helping or facilitating transport through. Another type of the passive processes here is going to be osmosis. Now, osmosis is defined as diffusion of water across a semi-permeable membrane. The important part here is water is moving. The aspect of being semi-permeable prevents something else from moving. So we have to have two things, water and at least one other component. The water can move, that component cannot, because of the semi-permeable membrane. And this is based on what's called osmolarity. Now, osmolarity, as it's said here, is the total concentration of solute particles in a solution. So if you're looking at this, you can have different osmolarities depending on different particles, different concentrations. But looking at what the concentration of either all the particles is, or it can refer to the osmolarity of a particular particle, a particular ion if you want to. So we have an example here. In this beaker, that dashed red line in the middle is going to be the semi-permeable membrane. You'll notice that on one side, we have a much higher concentration. On the other side is a lower concentration. Now, the liquid in here, the water, is able to cross the membrane, but the solute cannot cross the membrane. Again, we want to make sure that all of our environment is maintaining a homeostatic balance. So without any external factors, that means your body wants to make sure that the concentrations are equal on both sides. Well, right now, initially, this one that has the higher concentration has the higher concentration, more particles. So we have to make them even. The way that works, the water is going to cross the membrane, so it dilutes the side of higher concentration. By having the water cross over, you can see that both these now are roughly in the same concentration, about the same number of particles, and the spacing is about the same. The water moved across the membrane. And therefore, because of this semi-permeable membrane, you can see that the water levels are not equal. It's a way to force water to move across the membrane. So one of the ways our cells help to regulate their hydration, or the amount of fluid they have in the cells. The idea of osmosis. Now, in osmosis, we have something called osmotic pressure. Now, osmotic pressure, by definition, tendency to resist further water entry. Well, our cells have that phospholipid membrane. They can only swell up so far with water. So as the water fills into a space, eventually gets to the point where it really can't come in anymore because there's a lot of water there already. Now, this osmotic pressure goes right hand in hand with hydrostatic pressure. Hydrostatic pressure is the back pressure of water against the membrane. So what that's kind of referring to is as you bring more fluid in, it's pushing that membrane out. Well, the membrane wants to push back against that. It can only stretch so far. Usually osmotic pressure and hydrostatic pressure kind of go hand in hand. They're both trying to prevent further entry of water. Just the hydrostatic pressure is the pressure of the water itself. The osmotic pressure is the fact that there's not enough of the actual solutes to bring more water in. They're not having a high enough concentration anymore. And that brings us to the idea of tonicity. Tonicity is how a solution or how it is affected by cell volume. Because your cell volume needs to maintain a certain amount for the cell to be functional. If it shrinks down too much or blows up too much, the cell will not be very efficient. So we're always trying to maintain what's referred to as isotonic. Isotonic is the proper homeostatic balance the cell wants to maintain. So your red blood cells, for example, they want to maintain this shape. They want to be in about a 0.7% salt solution. That keeps them in the proper shape, the proper functionality. But if we end up in a hypertonic solution, so hyper means too much, tonicity as the actual concentration. So hypertonic means too many particles. This hypertonic solution means that the solution around the actual cell has a higher concentration than normal. Because the red blood cell, or any cell for that matter, has a semipermeable membrane, the water can leave. The water is going to try and leave to cause a dilution to occur outside, so you don't have the hypertonic solution. It's trying to lower the tonicity. Well, that's great to try and maintain homeostasis, but that results in the cell shriveling up because the water's leaving to try to maintain that balance, to make sure that the outside and inside are in equal proportions of tonicity. So you don't want the cells to be in a hypertonic solution. Likewise, you don't want the cells to be in a hypotonic solution. Hypotonic, hypo is low, so not enough particles are out there. In this case, if there's more particles inside the cell than outside, water flows into the cell. As water goes into the cell, that causes the cell to swell up. Again, not very efficient. And if this becomes too extreme, that cell that swells could actually rupture and die off. So we want to maintain an isotonic environment, not hyper or hypotonic, but isotonic. Filtration. Now, filtration is another one of the passive processes. Filtration is working off of hydrostatic pressure. Because in this case, we look at the filtration in the kidneys. That's a podocyte cell that's wrapping around a blood vessel inside the kidney. The blood wants to be filtered. It wants to push the particles out. So you're trying to make sure that you have enough pressure inside the blood vessel to force the particles out. There's little tiny slits here. It's allowing the filtration to occur. Particles get pushed through these little slits. Anything that's larger and can't fit stays in the blood vessel. So filtration is one method also of a passive process. So all these passive processes are not utilizing ATP directly. They're all utilizing kinetic or movement of energy a lot of times. They're utilizing pressures that are already established, like hydrostatic pressure, using osmotic pressures. So looking at tonicities, looking at even basically concentration gradients. But they're all utilizing things that are already present to allow for the movement of particles or ions. What we need to get to now is move into the active processes next.